Hello, fellow classmates. My name is Samira Remal, and my partner is Chelsea Killette. Today, we are presenting the topic of proanthocyanidins, PAC-A for short, in cranberries, decreases the frequency of urinary tract infections, UTIs, by the downregulation of E.coli genes. So to start off, we'll talk about the molecule itself. It's part of the flavonoid family. It's a class of polyphenols. It's known to have strong anti-adhesion properties. Also, it is to be noted that the double bond in the molecule, as indicated in the photograph below, circled in a red circle, is what allows for the characteristics of the anti-adhesion properties. And so that's why it is thought to decrease the occurrences of UTIs.
Also, next to the cyanidins, we have the E. coli and cell wall adhesion. There is a picture of the E. coli bacteria there. It is the primary bacteria responsible for UTIs. PAC-A downregulates these genes in E. coli. PAC-A is shown to decrease E. coli cell wall adhesion via downregulation of the BFGHI gene.
So next we have the project goal. We are investigating the validity, that is this prevention of urinary tract infections by PAC-A.
It's hypothesized that cranberry herbal supplements, regardless of dosage formulated, it won't significantly prevent the incidences of urinary tract infections or amount of bacterial flora when compared to placebo or other treatment options such as antibiotic treatments.
So next we have our research strategy. So here we just basically made a flow chart of how we're gonna tackle the issue of does PAC-A reduce urinary tract infections? So first we checked out some primary literature, some secondary literature, as well as some mechanisms of action. So the primary literature encompassed our clinical studies and our research studies, where we delved more into clinical trials. So actually seeing physicians and researchers working with patients, real humans, in different trials with amounts of cranberry. So then next we checked out research articles, which was a culmination of the primary literature. And lastly, we did see how gene regulation, either up or down regulation, affected pathways that went ahead and either supported or dissupported our hypothesis.
Okay, in the next column here, we have different figures representing our results here. The first one displays PAC-A in cranberries, which decreases bacterial flora and adherence via downregulation of many E. coli genes. We have panel A, B, C, 1, C2, and D. In panel A is mRNA, shown is mRNA expression of QRT PCR of genes involved in adhesion, motility, and biofilm formation, which are downregulated, and iron metabolism and stress response, which are upregulated. It shows the expression that is responsible after treatment with 190 micrograms per milliliter cranberry. In panel B, we have results showing genes upregulated and downregulated by cranberries, in which 1,245 genes were upregulated and 2,190 genes were downregulated. Panel C1 and C2 display fluorescence microscopy of E. coli cultured in urines, urines collected after cranberry consumption. C1 represents the E. coli culture in urines after placebo consumption, and in C2 represents the group that of volunteer that urine samples were collected 6 hours after consumption of 36 milligrams of PAC-A. Panel D displays E. coli in culture in urines collected 6 hours after consumption of cranberry powder, which contains 72 milligrams of PAC-A.
So next we went ahead and we have our pathway schematic shown. It's showing the interference of certain E. coli genes that is due to our PAC-A binding to the receptor. So first we have 5 networks that are affected: adhesion, motility, biofilm production, iron metabolism, and stress response. Over to the far left corner, we have our 5 nodes listed. Although there are only 5 nodes listed, there are nearly 3,000 genes that are actually affected by PAC-A. We went ahead and only list 5, and the ones that are down-regulated are color-coded in the pinkish red, and the ones that are up-regulated are color-coded in green. So we're going to focus on the ones that are down-regulated. So as you can see, adhesion is down-regulated due to fimbriae production being down-regulated, as shown in the pathway. So I'll point it out with my mouse. If you look here, these blue lines and these purple lines, they represent fimbriae. The blue is the type 1 and the purple is the type P. Now both of these genes here, this FMBFGH1 and the FLGAHK, are both downregulated, and they both downregulate adhesion. So we also have motility being downregulated, and that's being shown here. This big pink square is a flagella motor, and the gene responsible here is MOT-A and MOT-B, but it's combined in one, and that gene is downregulated as well. That downregulates motility. We have biofilm production being downregulated as well due to the exopolysaccharide production being downregulated by this gene, as well as chemotaxis being downregulated by this gene shown where I'm circling. Also, there is a blue box that is also pointing it out, that it's a gene responsible for chemotaxis. I would say that pretty much sums up our pathway. I would just want to finish with it. It supports the notion that PAC-A, it does downregulate genes that are important enough to actually downregulate the success of E. coli. Then next, we also have a figure here. It's 4-figured panel. It's basically just trying to show that A and B are showing the decreases in bacterial flora, and then C and D are just showing the decreases in adherence. And so there is some more information to the side that just goes to prove that we're seeing less amount and then less adherence.
And moving along to our third column here, just to reiterate, proanthocyanidins in cranberry, they may decrease the frequency of urinary tract infections by the downregulations of E. coli genes. These genes may influence proteins responsible for adhesion, motility, biofilm formation, iron metabolism, and stress response. The first three downregulate while the only downregulating the first 3 networks listed, PAC-A nearly affects 3,000 genes, maybe more. It was hypothesized that cranberry herbal supplements, regardless of dosage formulation, would not significantly prevent the incidences of urinary tract infections or the amount of bacterial flora when compared to that of placebo or other treatment options like antibiotic treatments.
So when we went ahead and we looked at the data, we showed that our hypothesis was not supported. It was actually the contrary, that PAC-A downregulates genes related to the fimbriae formation, like I discussed, as well as genes related to chemotaxis, like I had discussed in our pathway. And so this decreases the flora quantity as well as the adhesion, which is also shown in the figure that we talked about earlier. So this all goes to basically support the notion that PAC-A does decrease the frequency or possibly the adhesion of the E. coli responsible for the urinary tract infection.
Okay, for future studies, it is recommended that you complete clinical trials with more subjects with longer durations, also to decrease subject dropout rate by carefully selecting dosage formulation, and also running experiments with other types of bacteria besides E. coli for comparison reasons.
And lastly, we have our references and our acknowledgments, where we acknowledge the University of North Florida as well as Dr. Remal. Thank you all so